The lattice light sheet microscopy is a cutting-edge microscope which is built for the cell biologist. The system generates an optical lattice to create an ultra-thin light sheet to image biological samples over a long period of time and with very fine resolution. In this microscope allows for the 3D and 4D live cell as well as the fixed cell imaging. In the wide field microscopy, the entire sample is illuminated, giving the fast acquisition and easy to use. But what are the disadvantages? So it's a photo bleaching of entire sample, you can see, and no optical sectioning and lots of background. So the next is the confocal microscope. We will get the high resolution image and easy to use. The disadvantage, so of course the photo bleaching is the major problem, poor optical sectioning and slow acquisition. And so then here come the light sheet microscopy. This is also known as the Gaussian light sheet microscopy. So they use the cylindrical lens and they stretch the light, and then this stretched light will fall on the sample, and this is the detector. Orthogonally we will detect the light. So when they look on the picture, so you can see here and how to overcome with this problem. So what they got the idea here, instead of coming from one side, so the laser, so they put the two laser, one is left and one is right. They stretch the beam and then both beam are coming to the sample, and orthogonally it is getting the detection getting the orthogonally. So here this is the image how it looks like. It's much clear from the before. So what are the advantage of this microscope? It's a super optical sectioning, fast acquisition and low photo bleaching. What are the disadvantages? Here the Gaussian beam leads to trade-off between sheet thickness and the field of view. Less uniform illumination and due to that lower axial resolution. Let's compare these microscopes. So here this is a wide field microscope. The working principle is the full field illumination and detection. Advantage, it's a fast, simple and low cost. But it is giving the poor sectioning and out of focus light. Which kind of material we can use? It's a thin samples, fluorescently labeled cells or live cell imaging, but for the short time. So the next is the confocal microscopy. The working principle is the point scanning laser with pinhole to exclude out of focus light, and advantage is the high resolution image and good optical sectioning. So limitation is the slow photo bleaching and phototoxicity. It is good for the fixed cell, thick tissue imaging and 3D reconstruction. Next is the Gaussian light sheet. So here this side illumination with a thin sheet and orthogonal detection. It is fast, low phototoxicity and good 3D imaging. The limitations are that it requires sample mounting on in specific orientation. However, it is good for the live imaging of embryos, organs and clear tissue. So next is the lattice light sheet microscope. So here the working principle is the structured lattice light sheet for the thinner illumination. And advantage is it's a super resolution, very low phototoxicity, very fast and excellent for the 3D and 4D imaging. Limitation is it's a premium technology and that's why it's priced accordingly. So it is ideal use for the long-term live cell imaging, developmental biology and subcellular dynamics. This is the schematic diagram of the lattice light sheet microscopy. So using the three kind of laser, 488 nanometer, 561 and 642 nanometer. But however, in this setup, we can use some 6-channel laser together. So the light is coming here. So basically here we are using first the two set of cylindrical lens. This is the one set and this is the second, another set. So we are using the two set of cylindrical lens, and this cylindrical lens will stretch and collimate the beam. And then after that, this beam will go, here is the polarizing beam splitter, and then the certain polarization will go to the spatial light modulator, generate the optical lattice, and then here is the half wave plate. The light will come here like this, and this is the annular mask. It will generate the Bessel beam, and then finally this light will, here the light will interfere, and then this is the galvo, and galvo will dither the light. The lattice sheet is formed over the sample through the excitation objective. Here, this is the two cameras. They will capture the image through the detection objective. Here, this illumination and the detection object are both are the orthogonal to each other. And also, we should appreciate that this thin sheet of light excites only one plane at a time. That's why it is having the very less photo reach. So here it is a cylindrical lens. So this cylindrical lens have the special kind of lens. When the beam is light is coming, they will stretch this light. And this is the next is the spatial light modulator. So this is a liquid crystals basically. So if you apply certain voltage, so here how we are applying the voltage by this polarized light. So if you change the polarization, the voltage will be changed accordingly. And due to that, this liquid crystal will align, and then finally we will generate the optical lattice. So if you change this polarization, according to that the voltage will change, and then you in that way you can control this optical lattice. We have several optical lattices. And the next part is the annular mask. So this generate the Bessel beam. In this mask, there is some small holes, and all these are the small holes with the different diameters. Why do we need this? So basically, if the light sheet length is the 15 to 20 micrometer, then it will be helpful to understand the intercellular events. If it's a 30 to 40 micrometer sheets, then we can study the small organoids and the cell-cell interaction. And if it is 50 to 75 micrometer sheets, then it will be helpful the large organoids and the portion of the embryos or some large organism. So here, if the laser is coming, so laser is having this Gaussian mode, and this is the annular aperture. So here basically we are converting the Gaussian beam to the Bessel beam, because in this lattice light sheet we are using, because in our previous microscope we were using the direct laser light, so it will block some portion of the light and it will allow this rest of the portion, and then it will focusing, and then here you can see this is the Bessel beam. Here this is our interested area. This is the schematic diagram. So here you can see this is the only these 2 points. The light is coming through these points, and this is the range where this Bessel beam is where we are interested. This is the another view, that if the light is coming like this is the Gaussian mode, and this is the annular aperture. What we will get? We will get this kind of beam, and if you are focusing, we will get the Bessel beam like this. But here, let's say I am just giving you example. So if this is the laser is coming, some Gaussian beam, the laser is coming, and you are focusing. So laser always focus like it, and it is known as the ellipsoidal focus. So here you can see it's never be a point source. You can see this is the fundamental property of laser. It is, and this radius is known as a beam rest. As I mentioned, we can generate the Bessel beam by several ways. This is the axion prism. This is some special kind of prism. So here in this, if you are coming, and then in this region, it will generate the Bessel beam. If you are having the annular aperture, so you will getting the Bessel beam same way. The Bessel beam is known for the non-refracting, and it's a self-healing beam. Second, it has a longer propagation with thin central core, so that we can get more in a more depth. This is the table in the lattice light where which we can get in the 3i also. So here basically we are using the two objective. So one is the excitation objective, which is having the 0.7 and 71 Na and 3.7 working distance. And for the detection objective, it's a 1.1 Na and 2 millimeter is the working distance. In this, the magnification is 62.5x. I already mentioned that laser is 488, 561 and 642 nanometer. Here we are using the two type of camera. Both are the Hamatsu camera. All the sample is prepared with the 5 millimeter circular cover slip. We can control the temperature, we can control the carbon dioxide and the humidity level. These are the filters which we were using, and then some other specifications. Here, firstly, we have to do the daily alignment. So here, basically, we are looking the beam profile, the tilt correction, and we are looking on the aberration and the PSF correction on a daily basis. So this is how the 488 nanometer laser beam profile look like. This is the single bezel beam, and this is the multi-bezel lattice light sheet. Here you can see how the light is looking up, and this light we are going to observe, we are going to real measurement. This is the comparison. So here this is the mouse embryonic fibroblasts. So here you can see this image is the lattice light sheet microscopy, and this one is in the spinning disc confocal. So here you can see in this lattice light sheet how clearly we can see the dynamics, how clearly we can whatever the organ you want to look. ER and lipid dynamics in the live cell imaging of the COS7 cell. And here if you see, now this lipid droplet have a lot of dynamics and they are really interacting with the ER. So here this red thing is the mitochondria, which is tagged with the marker, and the green are the lipid droplets with the tag. So here you can see the interaction between like this area, how the lipid droplet is interacting with the mitochondria. So here basically they are looking the cluster formation of this P97 protein, which is tagged with GFP in the response of streptococcus pneumonia bacterial infection in A549 cells. So this is the same A549 cell, and here they have tagged this P97 protein with RFP and streptococcus pneumonia bacteria with the GFP. And here you can see how this bacteria is infected the cell. It is going inside. You can see very clearly. And this is the zoomed view. Here you can see this areas. Here, this is the work done in the laser scanning confocal microscopy. Here also first he has saw, you can see the cluster is visible here, and the same study he has done in this lattice light sheet. But why he have choose the lattice light sheet, you can see very clearly in the images also, that the imaging speed in this, it is quite slow if we compare to the lattice light sheet. Phototoxicity, of course, we can get the very low phototoxicity in the lattice light sheet microscopy, and the field of view is also big volume, has a larger volume. What is the practical consideration? So, of course, the first is the refractive index matching between immersion and sample medium. Color correction, anyway, we are using which kind of microscopy, we are aware that color correction is the major thing for the object, and the sample mounting. What are the application in research? So in the cell biology, we can use like we can study the organelle motion, actin, remodeling. In neuroscience, synoptic vesicle tracking, exon dynamics. In developmental biology, like a zebrafish embryos, etc. And in biochemistry, we can study the dynamic molecular interaction studies. So what are the major advantage of the lattice light sheet microscopy? So the resolution is in the lateral mode, it's a 230 nanometer, and axial is a 370 nanometer. If you are going to the SIM, it will be 150 and 280 nanometer. The temporal resolution is around 200 frame per second. Of course, it's a low photo bleaching, sequential excitation with multiple laser, 4D imaging, and volumetric imaging with sub-second frame rates. So in this, I just would like to conclude that the lattice light sheet microscope bridges physics and biology, allowing scientists to visualize dynamic biological process scales from molecular to embryos in unprinted details. Thanks to Professor Eric Pritchett for this wonderful microscope, which he got a Nobel Prize in 2014, and thanks to all of you.